Now, anybody that's been following my work knows that for at least the last 10 years, I have been screaming from the mountaintops that it appears from Scripture that the only candidates that we have to explain how the Nephilim returned after the flood were the three wives of Noah's three sons. So you can imagine my excitement when, as I was watching Brien Foerster's video, he goes even further to describe what they found in the DNA when they started testing these skulls. Now keep in mind, they're testing skulls in Peru. And keep in mind that we know from Scripture that Noah's Ark landed where? On Mount Ararat in Turkey, which is just southeast of the Black Sea. All right, now check this out.
Supposedly all of the ancient Peruvians from pre-Columbian times, their ancestry comes from haplogroup B. And the source of that is Southeast Asia, and that is consistent with migrating across the Bering Land Bridge 12,000 plus years ago, and eventually migrating to the coast of Peru. However, when we did our DNA tests, the results became quite intriguing. Here we have, once again, the 18-month-old baby Paracas skull. Its maternal or mitochondrial haplogroup turned out to be U2E1, source of which is the Black Sea and the Caspian. And then the skull on the right-hand side, its haplogroup turns out to be T2, which again goes back to Europe or Eurasia. And then this one, its mitochondrial haplogroup is H1. It's found through Europe, but the source area is the Black Sea and Crimea. So it's highly unlikely of migration of people across land to Siberia, across the Bering Land Bridge, and all the way down the Americas to the coast of Peru. It's more likely maritime emigration 3000 BC or earlier. So again, another DNA test. The one on the left-hand side, H2A. Source, the Black Sea and Crimea. And then a more extensive series of testing. Again, the blood or the haplogroup we're expected to find in terms of mitochondrial is B. But instead, what we got from 17 different skulls, U2E1, U2E, H1, H, J1B1. Then some examples did show up, 4 examples of haplogroup B, as in B4, B4, then H and R, and H and B5. So the majority show ancestry not from the coast of Peru, not from migration through the Bering Land Bridge, but probable migration from the Black Sea and Caspian Sea areas, because that's where the other largest elongated skulls in the world have been found. The 2 locations in the world, the Black Sea and Caspian, Crimea area, and the coast of Peru. And the haplogroups confirm that. So here are some elongated skulls found in the Crimea, Black Sea area. And there you can see, in the case of the one on the right, there's an extra suture line coming down the forehead. That is consistent with what we also find in different locations in Peru. That is not normal in Homo sapiens sapiens. And very recently, a baby was found about 2 years old in Crimea, shown here, with the skull the size of the torso.
How crazy is that? I admit it, it's circumstantial, but I find it rather interesting that conehead people just so happen to have DNA that just so happens to trace back to the general area where the occupants of the Ark got off. And speaking of genetics, there's no evidence, at least none that I've seen so far, that the coneheads were giants. In fact, they all seem to be of comparable size to people today, 6 foot tall or even shorter. Some of these people are really short. So if my theory is that the wives look like this, how do we end up with giants? Well, I started doing a lot of research on the subject of epigenetics and recessive gene traits and how things of that nature work. And what it comes down to is DNA is code.
DNA is the instruction manual for how to build life. From animals to plants to humans, it defines us all. Information is stored in DNA using 4 types of molecules, which come in pairs. We have billions of these pairs. DNA folds itself into paired packages called chromosomes, which are stored in the nucleus of cells. Each species has a different number of chromosomes. Humans have 46 chromosomes, which contain our genes. Genes act as instructions for molecules called proteins. In humans, genes vary in size from a few hundred DNA bases to more than 2 million bases, and we have an estimated 20,000 to 25,000 of these genes passed during reproduction. Each person has 2 copies of each gene, one inherited from each parent, and most genes are the same in all people except for a small number of genes being slightly different. These small differences contribute to each person's unique physical characteristics, varying from height, color, and personality traits. Some genes are dominant and some are recessive. Dominant genes are more often expressed as observable characteristics, while recessive genes are often masked by dominant genes. The recessive genes are expressed when inherited from both parents.
Right? I think most of us know this, that there's microscopic code in our DNA that give instructions for the overall structure, whether we're talking plants, animals, or people, right? That there are specific codes that manifest specific traits. And if they started messing around with the human genome 120 years before the flood, who knows what kind of concoctions that they created and what the outcomes were. We do have various depictions throughout the ancient world of animal-human chimera, but what about the people who look more or less like typical people? Maybe they have longer necks, or maybe they have elongated skulls with larger brain capacity, perhaps other cognitive abilities that we don't have today. And what if it's just as simple as turning some of these codes on or off to manifest a trait? I believe that's exactly what we're dealing with here. And when it comes to gigantism, I'm going to kind of demystify that for you here. We typically think, okay, angel mates with humans, supernatural thing, giants are the result. But I think if we kind of rein ourselves in and just start looking at this from a scientific perspective, I think we'll find that the whole issue of gigantism is not very complicated. It could be as simple as either turning off a growth inhibitor gene or removing the code that limits growth. And I would point for verifiable scientific evidence to support this idea. I would point to the ligers. I don't know if this website is still up. This was a screen capture from quite a number of years ago from the website. I think it was liger.com, talking about the hybrid ligers. A liger is a hybrid of a male lion and a female tiger. Now, one of the things that's interesting about this that they discovered is that what they call a growth inhibitor gene. You and I have a gene, a genetic code in our DNA that's told your body when to stop growing. For me, there was a gene that said Rob stops growing at 5'11" at about age 16. So when I got to that age, that's when I stopped growing, and I've been capped off at about 5'11" ever since. And what they discovered is that there's the growth inhibitor gene exists in the female lion and in the male tiger. So if you get a female lion and a male tiger, that's called a tigon. Tigons are hybrids, but they only get as big as a typical lion or a tiger. That's a tigon. And that's because in the female lion and the male tiger, you have the genetic code that caps off their growth process. However, when you take a male lion and a female tiger, creating a liger, there's no genetic code there that tells it to stop growing. So as a result, it just keeps growing until it dies, I guess, presumably. And so gigantism is not some big mystery. It can either involve the removal of the gene altogether or the turning off of the gene that controls growth. And so this is tangible genetic evidence that's observable and repeatable. They've made quite a number of these ligers. Interestingly enough, the ones they have here anywhere, with the exception of maybe Sinbad, are named after Nephilim. And if you look at this, look how big this creature is. Look how big that chunk of meat is. I mean, that chunk of meat's about half the size of this woman's head, and that's like a tic tac to this thing. I mean, that's tiny. How much meat do you have to feed this thing in order for it to maintain its metabolic rate? It helps you to understand why the Hebrew spies said the land the Canaanites were in devours itself, because you have to have enough food to sustain that larger creature. And especially if these things are just continuing to grow, nonstop until they die. So let's look at this topic a little bit more scientifically and address the issue of genetics and how they work. Here's a video clip from a documentary called Seeds of Death.
DNA is the inherited material of life, and it is where genes are structured. So we inherit half of our DNA from our mothers and half of our DNA from our fathers. And within the DNA are the genes that encode for all the structures of the body. So the DNA is like a blueprint. Just like an architect has a blueprint for a building, the DNA is the blueprint for all the structures of the body. So when a gene is switched on to function, the information within that gene is being used to manufacture a protein. And that protein then builds the structure of the body. And then proteins in the form of enzymes carry out all of the chemical reactions of the body that constitute the living organism. The process of genetic modification is a completely irreversible process. Once it has been carried out, it affects the reproductive cells of the organism. So any effect you have, for good or for bad, is then passed down through the subsequent generations of that particular organism. And that's true for plants and animals, indeed for any genetically modified organism.
Okay, so we saw that in the pre-flood world they were genetically modifying themselves. They're blending themselves with other species of animals, reptiles and plants even. Okay, so there was genetic manipulation that took place. And if that genetic manipulation makes its way into the reproductive cells, then it's going to get passed on. And so there's something you can do called a Punnett Square that would enable you to figure out, what the odds are of what kind of offspring you have. Why would it be that Ham had quite a bit of giant offspring and Shem had none? How could that be? And what I discovered as I was looking into genetics is that, at least in the thesis that I'm putting forth, is that even if the wives were 50-50 hybrids, as long as the male is 100% pure, you have a 50-50 chance of producing either more Nephilim offspring or 100% normal offspring. And the Punnett Square shows it. I created, women have 2 X chromosomes and men have an X and a Y. So let's say we have a 50-50 hybrid female, and she has one corrupt, I'll call it a nephilisome, one corrupt X chromosome. I'll call it a nephilisome. There's 4 potential offspring she could have, 4 possibilities. If she contributes her bad X and he contributes his good X, then you have a 50-50 Nephilim female. If she passes on her bad X and he passes on his good Y, then you have a 50% Nephilim hybrid male. However, if she contributes her good X chromosome and he contributes his good X, you get a 100% normal female. She contributes her good X and he contributes his good Y, you have a 100% normal male. So I would say that this is what happened with Shem. That in every case, it turned out, 50-50 chance. In every case, it turned out that his offspring were 100% normal. Japheth had sort of a mixture of the two, and so did Ham. But when it came to the Canaanites, it appears that you were always in this category here. So again, as long as the men are 100% normal with a Y chromosome that goes all the way back to Adam, okay, no fallen angel Y chromosomes, because only men have Y chromosomes. So since there's no female angels, when the angels mated with women and produced male offspring, that male offspring would have had would have had fallen angel male Y chromosomes. And so when you have that scenario with a fallen angel, you get a big mess here. So if there's multiple incursions, you get a wide range of problems when you do this out. These are the possibilities that you end up with, when you're blending corrupt angel seed with normal women. You get all kinds of problems here. Okay, so if this theory is true, then, genetically speaking, what would determine the outcome? I mean, if it's 50-50 chance, right? 50-50 chance you can produce either 100% normal or produce another 50-50 hybrid Nephilim. What would determine the potential outcome? Well, I believe Scripture tells us. In Exodus 34:7, he talks about the iniquity of the fathers, the sins of the fathers being passed unto the third and fourth generation, or generation. We see that Ham was doing something wrong, right? And, that would show something in his character, probably, that he was predisposed to perhaps doing things rebellious. And that being the case, we see that the sin of the father is passed down to the third and fourth generation. That's why it talks about that until the sin of the Amorites is complete. Israel was wandering in the wilderness till the sin of the Amorite was complete. Of course, they had to walk out their 40-year judgment for their own disobedience also, but that we have 4 generations of Nephilim filling up the land of Canaan while Israel is wandering around in the wilderness. I believe that this is what's happening here. This is what is determining the outcome of the 50-50, roll of the dice. And we see in Deuteronomy 7:9 that Yahuwah keeps covenant and mercy with those that love him and keep his commandments to what? 1,000 generations. So if you're obedient to him, you follow in his ways, then I think the 50-50 chance, who controls the outcome of chance, right? That if we have this potential genetic problem that could be passed down, well, I suggest that the problem is held back through obedience or released through disobedience. And there may be other factors too. As I was looking into all this, again, from a scientific perspective, looking at genetics, it could be that the science of epigenetics may help us to understand what's going on with the Nephilim also. Now, the clips that I'm about to show you, they weren't talking about Nephilim, okay? They're talking about just epigenetics and how that affects the genome. I'm taking, shall we say, creative license here and applying what they're saying to what I'm saying here. So think in terms of Nephilim as you watch these videos dealing with the science of epigenetics.
The human genome is like a Bible where everything was written down. The hope and the expectation was that once we had that book in front of us and all the letters, we could just read down the pages and we would understand how the body was put together.
They estimated that the human genome, the book of life, would contain around 100,000 genes.
And then when they started sequencing, they popped down to 60, and then it popped down to 50. And slowly it went down to a much smaller number.
30,000, 25,000. As the mapping drew to an end, it appeared that humans had about the same number of genes as fish and mice.
In fact, we found out that the human genome is probably not as complex and doesn't have as many genes as plants do. So that then made us really question, well, if the genome has less genes in this species versus this species, and we're more complex potentially, what's going on here?
So few genes didn't appear enough to explain human complexity. Even more startling, it turned out the same key genes that make a fruit fly, a worm, or a mouse also make a human. Chimpanzees share 98.9% of our genome. So what accounts for the vast differences between species? Might genes not be the whole story?
What is it that makes our cells different from one another? What makes them look and behave differently? What is it that makes a muscle cell, for instance, look different from a neuron? After all, these cells contain exactly the same DNA, but it's their epigenetic instructions that help tell them which genes to turn on and which ones to turn off. And with those different genes at play, these can become very different cells.
As epigenetic therapy takes off, so do the expectations for this new science. Many believe that a multitude of complex diseases, from Alzheimer's to autism, may have epigenetic triggers. Something that could control genes directly, switch them on or off. But how exactly did these tags go about silencing a gene? This odd strain of agouti mice provides a visual clue. Despite the difference in color and size, they're twins, genetically identical.
Mice contain a gene called agouti, which makes them obese and yellow and susceptible to diseases like cancer and diabetes. This gene and these traits can be passed down from generation to generation through DNA, so that an agouti mother will give rise to a fat, yellow, disease-susceptible offspring if that offspring contains the agouti gene. Now here's something interesting about the agouti gene. It can be turned off if silencing epigenetic marks accumulate around it. So, if a pregnant agouti mother is fed a diet which is supplemented with these silencing epigenetic marks, those marks will be chemically transmitted to the DNA of her embryo, where they'll accumulate around that agouti gene and effectively turn it off. Her embryo will maintain those marks, and so it will be born and grow up to be an adult mouse that's thin and brown and healthy. Even though this mother is genetically identical at the DNA level to both sets of these offspring.
So what switched the agouti gene off in the thin mouse? A chemical tag called a methyl molecule. Composed of carbon and hydrogen, it affixes near the agouti gene, shutting it down. Living creatures possess millions of tags like these. Some, like methyl molecules, attach to DNA directly. Other types grab the proteins called histones around which DNA wraps and tighten or loosen them to turn genes on or off.
And in simple terms, this contact can be thought of as hugging the DNA. And if these proteins hug the DNA very tightly, then it is hidden from view for the cell, and a gene that is hidden cannot be utilized.
These tags and others control gene expression through a vast network in the body called the epigenome.
Epigenetics literally translates into just meaning above the genome. So if you would think, for example, of the genome as being like a computer, the hardware of a computer, the epigenome would be like the software that tells the computer when to work, and how much.
What distinguishes cells is not their genes, but how these genes are switched on or off by epigenetics.
And as development unfolds, certain switches need to be thrown. Then you can think of it as a light switch. Switch on the gene, the light is shining, the gene is active, makes a cell do a certain thing. Or the light switch is off, everything is dark, that gene is off.
To turn off the overactive agouti gene, researchers gave pregnant mothers foods rich in vitamins like B12 or folic acid, from which they could make those methyl tags that silence genes. The change was small, the effect huge. Fat yellow mothers gave birth to thin brown pups, no longer prone to disease. Agouti mice revealed the impact of an epigenetic change, one that occurred without altering a single chemical letter in the agouti gene.
I knew that epigenetics existed. I knew that it was a controlling factor for DNA activity, whether genes are silenced or not. But to say that epigenetics would have a major role in disease development, so I had no concept for that. The fact that this could have such a huge impact and could explain a whole host of things we couldn't explain before.
The implications, if they apply to humans, are sobering. If the thousands of genes identified by the Human Genome Project symbolized the words in the Book of Life, it was the epigenome that determined how that book got read.
I think that's really sobering indeed. And again, this is real science, and I'm just applying real science to what I'm seeing in the scriptures, and I think it's a match. I think it's matching up. I think it's showing us how things are, how things may have happened. And what I do find interesting in what they just said is they were referring to DNA as the Book of Life, and I think there may actually be real truth to that. I do believe that the Book of Life is the genetic code of the things that God created good and very good, and that He created specifically man in His own image and His own likeness, and that our name is written in the Book of Life from the foundation of the earth. So genetically speaking, the things that He created are in the Book of Life. But when we start messing with genetic code, we create something that's not found in the Book of Life. And that's what we're going to talk about later, probably in the last session, about those in the book of Revelation whose names were not found written in the Book of Life from the foundation of the world. I'm of the opinion that we're dealing with genetic hybrids that were never meant to exist in the first place. Their genetic code is not in the Book of Life. They were created, engineered by man as a result of the information given to us by fallen angels. That's my opinion on that.